Okay, picking up where we left off in Chapter 18, this is Table 18.4 in your textbook, and what it's describing for you are some of the effects of an underactive and an overactive thyroid. So just start this material and know it. Bottom line, remember that thyroid hormone is responsible for elevating your basal metabolic rate, so it increases your body temperature, increases the rate at which you burn fat, and as a result, when you have oversecretion of thyroid hormone, you're going to tend to have less overall body fat. You're going to have a higher body temperature. Your heart rate and respiratory rate will be elevated, and you'll also see an increased intake of iodine. Remember, because that's a structural component of the hormone. You're going to see enhanced smooth muscle activity, so diarrhea is going to be frequent. And you'll also see a bulging of the eyes, which we call exophthalmia. That's an unusual side effect of this condition. And in hypothyroidism, you basically have the reverse of the bulk of these symptoms. Next hormone we want to talk about is calcitonin, and calcitonin has as its main effect upon secretion by the thyroid gland the reduction in blood calcium levels. It's made by the parafollicular cells, which are between the follicular cells, basically wedged between the follicles. Secretion is triggered by high calcium concentration in plasma, and it acts to reduce that concentration, and its primary target tissue is bone. And what it does is it decreases osteoclast activity, and it lengthens the lifespan of osteoblasts. Remember that the osteoblasts are the bone builders, the osteoclasts are the bone dissolvers. The parathyroids are on the back side of the thyroid gland at the edges. Average about 4 parathyroid glands per individual, though there is some variation in that number. They secrete parathyroid hormone, which has the opposite effect of calcitonin. It elevates blood calcium levels by increasing osteoclast activity. It promotes calcium reabsorption in the kidneys along with phosphate excretion, and increases the synthesis of vitamin D, which increases absorption of calcium and phosphate by the intestine, and net loss of phosphate as well. So the bottom line, we have to keep a relatively tight control on blood calcium because calcium is critical in many important physiological functions, among them blood clotting, muscle contraction, and nerve transmission. And so if we allow calcium levels to get too high or too low, all of those are going to be affected. This is just a look at how we regulate calcium levels. We can start here at the edge of the regulatory curve. Okay, blood calcium levels decrease, so the homeostasis is disturbed. So what's going to happen is parathyroid hormone is going to be secreted by the parathyroid gland. Basically, what happens is that because this area of tissue is highly vascularized, it can detect when calcium levels begin to fall. The effector is activated, so what happens is that the osteoclasts start to break down bone and release calcium into the blood. Increased absorption from the intestine is also seen, as well as more calcium retention by the kidneys, and the blood calcium levels begin to go back down. Okay, so what happened, or go back up. Now, if we start on the flip side, we're looking here at dropping or increasing calcium levels. So what's happening, blood calcium is beginning to rise. What happens now, homeostasis is disturbed. The result is going to be the production of calcitonin, and also a reduction in the secretion of parathyroid hormone. The result will be to remove calcium from blood plasma. Osteoblast activity will be enhanced. Osteoclast activity will be reduced. We're going to see increased bone density and increased uptake of calcium from the intestine and the kidney. and the result is that the levels are going to be restored. Okay, so the two hormones, of course, act to counterbalance each other. It's important that we understand, the activity in the skeletal system is relatively straightforward. We understand what the osteoblasts and osteoclasts do, but we also have to appreciate the fact that calcium absorption from the intestine is a major player in blood calcium levels, as is the activity of calcium retention at the kidneys. Okay, so keep those three systems in mind when we're thinking about parathyroid hormone and calcitonin to regulate blood calcium. And this is just sort of a list showing you what happens when the parathyroid glands misbehave. Hypoparathyroidism would be a lack of secretion of parathyroid hormone from the parathyroid glands, and hyperparathyroidism would be an oversecretion. And either one of these is going to result in profound effects on muscle contraction and nerve transmission. Okay, so make sure again and star that material and know it. The adrenal glands sit on top of each kidney, and there are actually two glands in one. There's an outer cortex, which is split up into three different layers, each of which produces a different steroid hormone, and then there's a core, which is nervous tissue, and it's actually an extension of the sympathetic division of the autonomic nervous system. When stimulated, it secretes massive amounts of adrenaline into the bloodstream. They are retroperitoneal, so they lie along with the kidney behind the peritoneum in the abdominal pelvic region, and the cortex three zones are known as the zona glomerulosa, the zona fasciculata, and the zona reticularis. And there's a relatively straightforward way to remember these three regions and what they secrete. If you go from superficial aspect to deep aspect of the cortex, the outer aspect is going to be the capsule, and that's not endocrine tissue. But then the glomerulosa is responsible for secreting the mineralocorticoids, the most important one being aldosterone. The fasciculata responsible for secreting the glucocorticoids, among them cortisone and cortisol, and the reticularis secretes androgens. So the way that I was always taught to remember this is salt, sugar, sex. So let's just write that up here in blue. Okay. So we have salt, and then we have sugar, and we have sex. Now you might well ask, what do each of these hormones do? Well, aldosterone, which is made by the glomerulosa, is our salt-retaining hormone, so it causes the kidneys to hold on to sodium and excrete potassium. The cortisone and cortisol, made by the fasciculata, elevate blood sugar levels and reduce inflammation and suppress the immune system. And the androgens are basically testosterone mimics, and so what they do is affect secondary sex characteristics and to some extent reproductive cycles. So in the adrenal medulla, the main hormones are epinephrine and norepinephrine. They combine with adrenergic membrane-bound receptors and all function through G-protein mechanisms. So GTP is involved on the cytoplasmic side of this signaling cascade. Secretion of the hormones prepares the body for physical activity, but the effects are short-lived and the hormones are rapidly metabolized. Epinephrine and norepinephrine are amino acid derivatives, and they're called catecholamines. Epinephrine increases blood glucose levels and increases fat breakdown in adipose tissue and causes dilation of blood vessels in skeletal muscle and in cardiac. Epinephrine and norepinephrine increase heart rate and force of contraction, so this increases cardiac output, and it also causes blood vessels to constrict in the skin, in the kidneys, in the GI tract, and in other viscera. So here we can see the cascade of events that leads to epinephrine secretion. Stress, physical activity, emotional upset, or low blood glucose levels will act to stimulate the hypothalamus, resulting in increased sympathetic nervous system activity. This increased frequency of action potentials is going to hit the medulla, causing it to secrete epinephrine and norepinephrine into the bloodstream. The epinephrine and norepinephrine act on their targets and produce their responses. So what are their targets and what happens? Well, it's going to increase the amount of blood glucose from the liver into the blood. So it spurs gluconeogenesis and glycogenolysis in the liver. It increases the release of fatty acids from the liver and from fat stores around the body. It increases your heart rate and your respiratory rate. It decreases blood flow through blood vessels of most internal organs. It increases blood flow through blood vessels of the skeletal muscle in the heart. It increases your blood pressure. It decreases the function of visceral organs, and it increases the metabolic rate of skeletal muscle. So here's just a
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit which dissociates from the others carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of cAMP from ATP. The cAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
So here's just a look at how epinephrine actually produces the effect on its target tissue. We'll look at its interaction with its receptor and the cascade that follows. Okay, if we look at the adrenal cortex, what we have to appreciate is that it's really 3 endocrine organs in one. It has 3 distinct zones that look histologically quite different from each other under the microscope, and the hormones that they produce have extremely varied function. So we start with the zona glomerulosa. The main steroid hormones produced here are the mineralocorticoids, the most notable one being aldosterone, which has its effect the retention of salt by the kidneys and also excretion of potassium. This also has an indirect effect of causing the body to retain water. The glucocorticoids are the purview of the zona fasciculata, the middle layer of the cortex. Cortisol is the major glucocorticoid. It increases fat and protein breakdown. It increases glucose synthesis and decreases the inflammatory response. So its overall effect is to suppress inflammation and elevate blood glucose levels. And then finally the androgens, which are produced by the zona reticularis, and they mimic the action of testosterone on peripheral tissues. So they stimulate secondary sex characteristics. They improve sex drive, and they also have the indirect effect of increasing muscle mass and bone density and hematocrit. So this is just a look at how cortisol secretion is regulated. So remember that we're talking a hierarchy that involves both the hypothalamus and the pituitary gland. So corticotropin releasing hormone is generated by the hypothalamus in response to stress or low blood glucose. It passes into the hypophyseal hypothalamic portal system to the tissue of the anterior pituitary, where it stimulates the production of adrenocorticotrophic hormone. This then enters the bloodstream and binds to membrane-bound receptors in the adrenal cortex, stimulates the production of glucocorticoids such as cortisol, which will then act on its targets, promoting fat and protein breakdown, glucose synthesis, and its anti-inflammatory effect. Cortisol, as it builds in the bloodstream, will have a negative feedback on both the hypothalamus and the anterior pituitary. The result is that the initial command will be turned off or turned down, and the levels now of cortisol will drop back to normal under normal conditions. If stress levels remain high for an extended period of time, what eventually will happen is that the amount of cortisol in the bloodstream, along with epinephrine, is going to have an adverse effect on our overall health. Our blood sugar levels are going to start to climb. We're going to have suppression of inflammation and immune response, and eventually this is going to bring on disease. So this is just a look at how glucocorticoid hormone does its trick.
Glucocorticoid hormones are steroid hormones that influence nutrient metabolism. These hormones activate genes for proteins involved in processes such as the synthesis of glucose, the breakdown of proteins, and the mobilization of fat. Glucocorticoid hormones are secreted by endocrine glands into the bloodstream. They enter cells through transporters in the plasma membrane. Once inside the cytosol, the hormone molecules can bind to glucocorticoid receptors, which function as transcription factors that activate genes. The binding of the hormone causes proteins that were bound to the receptor, such as the heat shock protein HSP90, to be released. Release of HSP90 allows two receptors to bind and form a dimer, as well as exposing a nuclear localization signal on the receptor. The receptor dimer is then allowed to enter into the nucleus through a nuclear pore. The receptor dimer is able to bind to specific DNA sequence regions called glucocorticoid response elements, or GREs, that are near target genes. The GREs function as enhancers, so that binding of a receptor dimer to a GRE activates transcription of the nearby target gene.
And this is a look at steroid hormone activation on its targets. Remember that steroid hormones are nonpolar, so their receptors are actually inside the target tissue, and they are going to turn on the production of specific gene products. So let's take a listen.
Steroid hormones are not water-soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins, not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
And this is just a table showing you the hormones of the adrenal glands. So you can see here the medulla and the cortex, the structure of the hormones that they produce, their target tissues and their response. This is 18.7 in your book. And this is 18.9, and this just addresses symptoms of hypo and hypersecretion of adrenal cortical hormones. So just star those two charts. Again, know that material. It's good PRS quiz material and exam material. Okay, the next endocrine organ that we need to address is the pancreas, which has a dual function. It's both an exocrine organ, where it acts primarily to generate digestive enzymes that break down protein, lipids, and carbohydrates, and also bicarbonate to neutralize the acid coming from the stomach. But its endocrine role is to regulate blood glucose levels by the production of two hormones that are similar in structure but opposite in effect, that being insulin and glucagon. It's located between the spleen and the duodenum and behind the stomach and behind also the peritoneal membrane. It is both an exocrine gland, as we indicated. It produces those pancreatic juices that contain a lot of the enzymes that break apart the macromolecules in our diet, ultimately with the goal of being able to absorb them into the bloodstream. And its endocrine role is carried out by clusters of tissue that are highly vascularized called islets. There are both alpha and beta islet cells and a few other cell types in the islets as well. But that's where the secretion of insulin and glucagon takes place. They're composed of the alpha cells are the glucagon secretors, the beta cells, the insulin secretors, and then there's another hormone that's less prevalently discussed when we talk about the pancreas, and that's somatostatin, and its action is opposite that of HGH. It slows growth. So this is just a look at the location of the pancreas, and this is a slide of pancreatic tissue. The islets are indicated here, and the surrounding tissue is exocrine tissue. The juices from the exocrine cells eventually make it into the pancreatic duct, which is going to empty into the duodenum. However, the secretions of the pancreatic islet cells go directly into the bloodstream. This is a rundown of the pancreatic hormones and the cells that produce them and the response, and also the target tissues. So this is 1810 and 1811 in your manual. So just go ahead and star that, know that material. Let's talk a little bit about how it is that we regulate blood glucose levels. So let's take as an example, okay, we'll start again at let's say a blood glucose level that would be beginning to rise. Okay, so blood glucose level begins to increase. This disturbs homeostasis. The result is that the pancreas is going to secrete insulin. What will happen is, as the insulin levels increase in the blood, that will be carried to target tissues. In the intestine, the digestive hormones, gastrin, secretin, CCK, they stimulate insulin secretion, and autonomic nervous system via the parasympathetic leads stimulate the production of insulin. So there is a little bit of communication between the nervous system and the adrenal cortex. The effectors are activated, so insulin stimulates glucose uptake by most tissues in the body, and it promotes glycogen storage in skeletal muscle and liver, and excess glucose is then converted to adipose tissue. So essentially what insulin does is the hypoglycemic hormone. It tends to induce a reduction in blood sugar either by storing the simple sugar as fat in subcutaneous or in the omentum, for instance, or in other areas as well. And it also is going to spur the uptake of glucose for fuel by cells that need it for metabolic activity. Okay, so it's a signal to cells in the body that there's food in the area and it should be used. As the blood glucose levels begin to drop, what happens is that we approach now homeostasis. and everything is back where it should be. If we look at the other side of this blood glucose homeostasis diagram, let's say that our blood glucose levels are beginning to fall below normal. What will happen is instead the alpha islets will secrete glucagon, and the result will be the production of glucose from non-carb precursors by the liver. We call this gluconeogenesis. That literally means to make new sugar. And also will promote glycogenolysis. That's the breakdown of glycogen stores in muscle and in liver tissue. Some of the stimulators, pancreatic islets, will detect the change in blood glucose and will secrete the glucagon in order to, again, elevate blood glucose levels. So this is not a hypoglycemic hormone, but a hyperglycemic one. We will see in the intestine inactivity of the intestine and decreased insulin secretion, and autonomic nervous system sympathetic stimulation of the pancreas will inhibit insulin secretion and cause glucagon to be secreted at a higher rate. The effectors are going to be essentially liver and muscle. Okay, liver is going to be spurred to produce glucose from non-carbohydrate precursors. That's what the gluconeogenesis is about. The glycogenolysis will simply cleave glucose monomers off of the complex carbohydrate glycogen, which is also known as animal starch, and that will elevate blood glucose levels. What happens in addition is that decreased insulin results in decreased glucose uptake and increased glycogen breakdown, as we pointed out, in liver and in skeletal muscle. So these two hormones are very important in controlling blood glucose levels, along with, we should also note, the glucocorticoids and epinephrine. So, if we look at the digestive system as a signaling mechanism for the pancreas, what we have to appreciate is that we have two states. We have what we call the post-absorptive state and the absorptive state. In the absorptive state, what's happening is that the body's being signaled that there's fuel in the blood and it needs to be taken up either and stored as fat or used for energy. In the post-absorptive state, essentially the body is between meals and we have to mobilize these fuel stores for fuel. And so we would do is generate glucose, put that into the blood. So soon after a meal, blood levels of nutrients like glucose and amino acids go up. The nutrients enter the blood, and parasympathetic stimulation also increases blood glucose levels and causes increased insulin secretion by the pancreas. And this is just a response to the elevated glucose levels. It's an attempt to bring them back down towards what we would think of as the middle of the normal range of blood glucose. The increased insulin secretion causes nutrients to move into cells. The nutrients move into the cells, and in a couple of hours after a meal, nutrient levels in the blood decline. The lowered blood glucose levels and increasing sympathetic stimulation cause insulin secretion to eventually drop. This happens in most cells, where what will happen is glucose uptake will decrease and switch back to lipid and protein breakdown. In addition, lower blood glucose levels and increasing sympathetic stimulation cause elevated glucagon secretion, and this will spur either the production of new sugar from fat or the cleaving of glycogen and the release of that fuel into the blood. Lowered blood nutrient levels also promote growth hormone secretion. That, remember, is an anterior pituitary hormone. And lower blood nutrient levels will also promote cortisol secretion. You can see here the effect on the liver is to cause it to release glucose, ketones, and triglycerides into the circulation. The effect on adipose tissue is to release fatty acids into the circulation, and glucose uptake will decrease as other cells switch to other forms of fuel in order to generate ATP. So this all happens after the insulin spike. Okay, so once the nutrient levels begin to decline, it's time for glucagon to take over and to promote these changes in its targets. In addition to that, keep in mind that growth hormone and cortisol are involved in this in indirect fashion. This is just a little video showing you how blood sugar regulation in diabetics initially is in an uncontrolled state and how we manage it medically. Keep in mind that there are two major types of diabetes: type 1 diabetes, in which insulin production is ceased because the destruction of the beta islet cells as a result of an autoimmune attack. And then there's type 2 diabetes, which is insensitivity to your own insulin as a result of essentially flooding the system with the insulin signal too long and too frequently and reducing the receptor concentration on the target tissues. So let's take a listen.
As blood glucose levels increase after a meal, non-diabetics release insulin. Insulin signals the cells in muscle and liver to take up glucose from the blood. This causes blood sugar levels to return to normal. In untreated type 1 diabetes, no insulin is released and blood sugar levels remain high. Type 1 diabetes is treated by injection of exogenous insulin. This insulin has the same effect as normal insulin. In type 2 diabetes, insulin is released in response to a blood glucose increase, but the cells do not receive the signal. One treatment for type 2 diabetics is Avandia, an oral thiazolidinedione that increases cell sensitivity to normal insulin. An effective treatment for obese patients is metformin, an oral medication that blocks liver synthesis of glucose.
Okay, this is just a look at how blood nutrient levels during exercise will change. We can start here on the left-hand side. When we exercise, what happens? Well, we're going to have a higher demand for nutrients because we're performing activity, so we need to generate energy. So sympathetic stimulation increases epinephrine secretion from the adrenal medulla and glucagon from the pancreas, and it also inhibits insulin secretion from the pancreas. So what happens is that our blood sugar levels are going to begin to climb, and that's in order to get fuel to the muscle so it can do its job. The epinephrine increases the rate at which glycogen in muscle cells is broken down to glucose. It also stimulates the production of new sugar from non-carbohydrate precursors, and that's the gluconeogenesis. So epinephrine and glucagon together increase glycogen breakdown to glucose molecules in the liver. The glucose molecules are released into the circulatory system along with gluconeogenesis, where we're synthesizing fresh monosaccharide from fat precursors. The epinephrine and also sympathetic stimulation from the autonomic nervous system increase the breakdown of lipid and the release of fatty acids from adipose. The blood glucose levels are maintained for normal nervous system function. And so this is essentially in sort of a long way around saying that when our activity levels go up, what will happen is that nerve stimulation and also production of hormones that increase blood sugar levels will go hand in hand to fill the energy debt that's being created as a result of the activity. Okay? As exercise happens for a longer period of time, in prolonged exercise, we see increased adrenocorticotropic hormone and growth hormone from the anterior pituitary. ACTH stimulates increased cortisol secretion from the cortex. That's going to elevate blood glucose even more. This increases protein breakdown in muscle and liver to amino acids and increases glucose synthesis from these amino acids through a variety of metabolic pathways, the two major ones being transamination and deamination. There are certain amino acids that can be converted to alpha keto acids, and those serve as the building blocks for fresh sugar. In addition to that, some components of fat are used to synthesize sugar, the main component being glycerol. Cortisol increases the breakdown of lipid and the use of fatty acids by muscle cells, as an energy source, and it slows the breakdown of protein and helps conserve them. This results in increasing the dependence on fats as an energy source. And the reason for this is that the body doesn't want to go straight to protein for energy. Protein is primarily used to generate building blocks for new protein. We want our energy stores to come primarily from fats and from glucose. If we have to use protein for energy, that's often what happens when we're in a starvation scenario. Blood glucose levels are maintained for normal nervous system function. Remember that sugar is brain food. That's the best way I know to keep that in your head. And the body becomes increasingly dependent on fat and protein for an energy source, more fat than protein. Okay, the protein indirectly by conversion to alpha keto acids. But remember that basically the protein serves as building blocks for anabolic processes. Okay, the hormones of the reproductive system are shown here in Table 1812. We list the testicular and ovarian hormones, their structure, their target tissue, and their response. So star this. This is good, again, PRS quiz material. The next gland that we want to talk about is the pineal gland, which is an unusual gland. It's on the other side of the brainstem from the pituitary. It's actually part of the epithalamus, and it produces melatonin and arginine vasotocin. Melatonin is more well understood, but even at that, the role of melatonin is a little hazy. We think that it has to do with circadian rhythm and may play a role in reproductive cycles. Light entering the eye stimulates neurons in the retina to fire action potentials, which are transmitted to the hypothalamus. These action potentials run from the hypothalamus through the sympathetic division of the ANS to the pineal gland. A decrease in light results in increased sympathetic stimulation of the pineal gland and increased melatonin secretion. An increase in sight results, or not in sight, in light, my mistake, results in decreased sympathetic stimulation of the pineal gland and decreased melatonin secretion. So the way to remember this is that as it gets brighter out, the amount of melatonin drops. As it gets darker out, the amount of melatonin increases. Melatonin inhibits gonadotropin-releasing hormone secretion from the hypothalamus and also may help regulate our sleep-wake cycles, or what we also know as our circadian rhythm. And the reason that we believe this is because of the cycling of this hormone between day and night. Some other hormones and chemical messengers include the thymus gland, which is in front of your heart and behind your sternum, and also certain tissues in the gastrointestinal tract. The main hormone produced by the thymus is thymosin, and it promotes immunocompetence in the T cell population. In the GI tract, there are hormones that regulate digestion and enzyme secretion, but we'll touch on those when we actually get to the GI. Other hormone-like chemicals include autocrines, which are signals released by a cell that affect the same cell. Essentially, what happens is chemical mediators, such as those that regulate the inflammatory response. These include modified fatty acids, such as eicosanoids and prostaglandins and prostacyclins, are produced as a response to injury or disease. Remember that the overall thrust of inflammation is to bring blood to the area. With blood come cells that are going to fight pathogens and repair damaged tissue. In addition to that, the inflammatory response is a warning not to use the affected area and to treat the injury. Paracrines are chemical signals that are released into the intercellular fluid and affect nearby cells. Endorphins and enkephalins modulate the sensation of pain. They bind to special receptors called opioid receptors. They have a shape but not a chemical structure that is very similar to compounds such as opium and morphine and cocaine. As a result, they have a pain-killing effect. What happens is that they anesthetize ascending pain signals that come from peripheral tissues. And there are also several growth factors that are thought to be paracrine as well. Now, as we age, we see a decrease in the secretory activity of certain glands. Growth hormone decreases as people age, except in people who exercise regularly. Melatonin begins to decrease in the amount that is secreted by the pineal gland, and the thyroid hormones and renin also see a decline. There's also a familial tendency to develop type 2 diabetes as a result of inadequate production of the insulin signal. And you could think of this as the beta islets dying, but it also has to do with decreased sensitivity of the target tissues to the insulin that's secreted into the bloodstream by the beta islets. Okay, that brings to a close this segment of Chapter 18. Again, remember to study this material as it will come up on the next PRS quiz. And I'll see everybody in class. Thank you.